Welcome to AP Biology Teaching Tips. This is Joy Killow. In teaching tip number 6, I'll share models I use to introduce cell signaling. In this lesson, there'll be an overview of communication, including basic categories, and an introduction to cell signaling. To prepare to teach this topic, open the AP Biology Curriculum Framework on your computer. You can find this PDF at AP Central. Next, search the term cell signaling. The first entry is under Big Idea 3. Living systems store, retrieve, transmit, and respond to information essential to life processes. Store and retrieve are mainly concerned with DNA and RNA, but transmit and respond involve communication and cell signaling. Cell signaling is mainly addressed in Enduring Understanding 3D. Go through the curriculum framework to review the depth and breadth for this topic. Signaling has some similarities across kingdoms and domains, illustrating its evolutionary commonality. A great resource is Bonnie Bassler's TED Talk on quorum sensing in bacteria. Bacteria use chemical messengers to communicate with other nearby cells and to regulate specific pathways in response to population density. This is a great introduction to cell signaling, but is also valuable near the end of the lesson to provide an aha moment as students put the pieces together. Organisms must communicate with other organisms within their own body and receive information from the environment. Communication allows coordination between organisms, as in these yeast cells sending and receiving signals that lead to mating. And within organisms, communication can be cell to cell over short distances, like this local signaling, or long distances, as we see with hormones traveling in the bloodstream. Immune system cells communicate by cell-to-cell contact, as seen between this antigen-presenting cell and helper T cell. Regardless of where the signal comes from, it must be received by the cell and acted on. This is signal transduction. Signals can stimulate or inhibit. You will need several models for the next activities. The cell signaling templates, seen here, the cell signaling placemat, seen here, and model pieces from the action potential model. All of these are available at this URL. You may recognize these pieces from the action potential model. This was seen in teaching tip number 4 and is the neurotransmitter acetylcholine crossing the synaptic cleft to a muscle cell. This is an example of a signal moving a short distance. Students can follow instructions on this slide, answering the questions, then modeling as a group. It's okay to use references. The string represents the vesicle membrane. The ligand is the acetylcholine. The interaction of the ligand with the receptor causes a shape change that opens a passageway or gate. After working through the last slide, you can allow groups to self-check, then write the actions in their journal. The nature of signaling depends upon the properties of the molecules involved. If you have used the colored models for macromolecules, your students may remember orange represents protein and green represents lipids. Before continuing, ask them to explain events in these two pictures as best they can. They need a what, but more importantly, a why. This is best done in small groups where students can offer support in a less stressful setting. This slide offers a checkpoint. Large and polar hydrophilic substances can't pass through the membrane. And another checkpoint slide for the hydrophobic molecules. The examples given are important. This model allows a deeper understanding of the molecules like testosterone. Ask student groups to model and share. Each molecule fits within the next. This video shows the hormone binding with a transport molecule to cross the membrane, and then it binding to its receptor. This complex interacts with the DNA to turn on transcription. The mRNA is translated by the ribosome. This clip shows the construction of the puzzle model. This puzzle is also a good place to review transcription and translation. These puzzle pieces have been color-coded to meet the macromolecule coloring that's used in the rest of the models. A specific receptor that picks up molecules on the outside of the cell and passes the message inward is the G-ProteinCoupled Receptor, or GPCR. Though there are many different versions of the receptor, G-protein, and effector, adenylyl cyclase here, they work the same way. Note that the ATP model piece reverses to cyclic AMP. As with the other models, have students try their hand at this before you give them too much information. Think of your input as refinement. After a bit of effort, offer this script as a scaffold. You can pause this video here to spend more time with this script. Here's another scaffold that might help students. And one less scaffold. Shape change. G-protein translocates to the GTP, interacts with the adenylyl cyclase. ATP is converted to cyclic AMP, the second messenger. Ask students to model the amplification cascade before showing this clarifying slide. This lesson covers many learning objectives as seen here. Refer back to the curriculum framework to be sure you're covering all aspects of each learning objective. This will set the students up for looking at changes in cell signaling caused by mutation and disease. Assessment can be very aligned to the lesson if students make a video for one of the covered topics. Good luck with cell signaling. Until next time, this is Joy Killow and today's teaching tip.